Meiosis is the process in which gamete cells reproduce sexually. In meiosis, cells go through two interphases and two prophases, metaphases, anaphases, and telophases, and also cytokinesis. The point of all this is to create 4 genetically diverse haploid cells from only one diploid cell.
In interphase 1, the cell grows and functions normally. During this time, a high amount of protein synthesis occurs and the cell grows. More organelles are produced and the volume of cytoplasm increases. In prophase 1, the exchange of DNA between paired chromosomes via the process called homologous recombination occurs, and the crossover at chiasmata between the non-sister chromatids. Thus, this stage is important to create genetic variation. In metaphase 1, the homologous pairs of chromosomes align on either side of the equatorial plate. Anaphase 1 begins when the homologous chromosomes separate. The nuclear envelope reforms and the nuclei reappear. The chromosomes coil up, the nuclear membrane begins to disintegrate, and the centrosomes begin to move apart. Spindle fibers and the sister chromatids align to the equator of the cell. During telophase 1, the chromosomes are enclosed in the nuclei. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into 2 daughter cells. Each daughter cell is a haploid and only has one set of chromosomes, or half of the total number of chromosomes of the original cell. Cytokinesis is the process by which one cell physically separates into 2 daughter cells at the end of each division cycle.
Starting in interphase 2, there are no 2 haploid cells, and in interphase 2, the only significant thing that happens is that they create new organelles. In prophase 2, the nucleus is disbanded and spindles form, along with the centrosomes still being present, although there is no crossing over, like the first prophase. Metaphase 2 is very similar to metaphase 1 in the way that the chromosomes line up in the middle and attach to the spindle, just as they do in metaphase 1. Nothing is really different except for the alignment of these chromosomes can be in any way. In anaphase 2, the chromosomes are being pulled apart back into chromatids. They've done this by attaching to the spindle and being pulled apart to either side of the cell. Telophase 2 and cytokinesis are very similar to the first telophase and cytokinesis. New nucleuses are formed once they split, along with the cytoplasm being doubled and split into both. The centromeres are double and are still there. Along with this, the spindle is dissolved. In the end, we end up with 4 unique haploid cells. These could be sperm or eggs, but the main part is that they're all genetically diverse, none of them having the same DNA or code within them, so they all turn out to be different things, which creates genetic diversity among organisms that reproduce sexually.